The surface of the ocean moves with rhythm. Waves rise and fall. Light breaks through the water in rays that shimmer and fade. Everything you see is familiar, measured, knowable. But that familiarity is a kind of blindness. Beneath the visible world is another realm entirely. Not another ocean, but the same ocean transformed, where light becomes memory, where warmth drains away, where pressure builds until the water itself feels solid. You stand on a beach and watch the horizon. The line between sky and sea seems absolute, but it is only the beginning of distance. The ocean does not end at that line. It falls. If you could walk down from the shore into deeper water, you would pass through layers, each one darker than the last, each one colder, each one more remote from the sun. At 200 meters, the light turns blue and dim. At 600 meters, only faint traces remain. At 1,000 meters, darkness becomes absolute, and the ocean continues falling. 3,000 meters down, 5,000, 7,000. The average depth of the ocean floor sits near 4,000 meters, but trenches cut deeper still, 11,000 meters in places. Valleys that could swallow mountain ranges. Spaces where the weight of water above reaches pressures that would crush steel. And yet, even there, life exists. Everything you understand about living things comes from a single assumption. That energy begins with sunlight. Plants capture light. Animals eat plants, or they eat animals that ate plants. The chain always traces back to the surface, to photosynthesis, to the sun. This is not a theory. It is observation repeated endlessly. It feels as fundamental as gravity. But in the deepest parts of the ocean, that chain breaks. Sunlight does not reach the bottom. It cannot. The water absorbs it completely long before. And yet life does not disappear. It changes. It finds another way. A way that does not require light. A way that operates on principles hidden from the surface world. This is not adaptation in the usual sense. This is replacement. A different foundation entirely. What you are about to hear is not speculation. It is documentation of ecosystems that exist right now. Beneath your feet if you stand near any coast. Beneath every ship that crosses open water. Places where chemistry replaces sunlight. Where heat rising from the Earth's core becomes the source of everything. Where entire communities thrive in conditions that would extinguish most life instantly. These are not rare anomalies. They are vast, populated, ancient, and they have been operating in complete darkness for as long as oceans have existed. This is the story of life without light, not as theory, but as fact, observed, measured, ongoing. Welcome to Info Jungle. If you enjoy calm, thoughtful documentaries about the natural world, consider subscribing. These videos are created to help you relax while learning at a gentle pace. Begin with what is known. The surface of the ocean teems with life. Phytoplankton drift in the currents. Microscopic plants too small to see individually, but visible from space when they bloom in billions. They float in the sunlit zone, the top layer of the ocean, warmed by the sun, lit by the sun, fed by the sun. These tiny organisms perform photosynthesis. They take in carbon dioxide, They take in water, they take in light, and they produce sugar, glucose, energy stored in chemical bonds. This process powers nearly everything. The fish that swim through reefs, the whales that migrate across oceans, the seabirds that dive for food. All of them depend on photosynthesis somewhere in their food chain. Even predators that never eat plants consume animals that did, or animals that ate animals that did. The chain connects everything to light. This is the familiar pattern, the one taught in schools, the one that seems universal. And for most of the ocean's surface, it is. But the surface is only a thin skin, a bright layer spread across the top. Below it, the ocean changes. The transition happens gradually at first. As you descend from the surface, the water grows darker, cooler, less populated. Light penetrates to different depths depending on its color. Red light disappears first, absorbed within 10 meters. Then orange, then yellow. By the time you reach 100 meters down, only blue light remains. Dim, scattered, fading. This region is called the twilight zone, not because of any mystery, but because of the light itself. Enough to see by, but not enough to sustain photosynthesis at any meaningful scale. Plants cannot grow here. The light is insufficient, yet life persists. Animals swim through this dim layer. Fish with large eyes adapted to low light. Squid that migrate up and down with the day and night cycle. Jellyfish drifting in the currents. They feed on the debris falling from above. Dead plankton, fecal matter, fragments of larger creatures. A rain of organic material that sinks slowly through the water column. This is called marine snow, not because it resembles snow, but because it falls like it, constantly, silently, feeding the deeper layers. The animals here do not create energy. They recycle it. They consume what was made at the surface. They are still connected to the sun, just indirectly, at an increasing distance. At 200 meters, the twilight zone deepens. At 400 meters, only the faintest blue glow remains. At 600 meters, a human eye can detect almost nothing. At 1,000 meters, darkness becomes total, absolute. No light from the surface can penetrate this far. The water absorbs it completely, scatters it, converts it to heat. This is the midnight zone, named for the permanent night that exists here. Not metaphorical night, not temporary night, but a darkness that has persisted since the oceans formed. For 3.5 billion years, no sunlight has ever reached these depths. And yet they are not empty. Fish live here. A strange fish. Anglerfish with bioluminescent lures dangling in front of their mouths. Viperfish with teeth too large for their jaws. Hatchetfish with bodies flattened like blades. They hunt each other in the dark, using senses beyond sight. Detecting vibrations in the water, sensing chemical traces, producing their own light through chemical reactions in specialized organs. But they do not create energy. They consume it. They eat other fish, or they eat crustaceans, or they eat the marine snow falling from above. All of their energy still originates at the surface, filtered down through hundreds or thousands of meters of water, pass from corpse to corpse, from predator to prey. The sun still powers them, just at a great remove, through many intermediary steps. They are darkness dwellers, but they are not independent of light. Not yet. Below the midnight zone, the ocean floor begins to appear. Not everywhere. The ocean is not uniform. In some places, the bottom sits at 2,000 meters. In others, 5,000. In the deepest trenches, it drops beyond 11,000. But wherever the floor exists, it marks a change. The marine snow that has been falling from the surface finally settles. It accumulates, a soft sediment of organic debris, mud made from the remnants of surface life. Creatures live in this mud. Worms burrow through it. Sea cucumbers crawl across it. Brittle stars drag themselves slowly over its surface. They feed on the settled material, digesting what remains of phytoplankton and zooplankton that died far above. The food is sparse, nutrient poor, already consumed and excreted multiple times during the long fall. Life here is slow. Metabolisms run cold. Growth takes years. Reproduction happens rarely. But it happens. The ecosystem functions. Quietly, patiently, sustained by the faint trickle of energy from the distant surface. This is the abyssal plain. The deep sea floor. Flat, dark, cold, covered in sediment, populated by animals that have never known light. And for a long time, this was believed to be the deepest form of life, the final step in the energy chain, where the last traces of sunlight-derived nutrients get consumed, where the system finally runs out. But in the early 1970s, that understanding changed. Not gradually, not through theory, but through direct observation. A discovery that revealed entire ecosystems operating on principles completely separate from photosynthesis. In 1977, scientists aboard a submersible called Alvin descended to the seafloor near the Galapagos Islands. They were studying the mid-ocean ridge, a massive underwater mountain range where tectonic plates pull apart, where magma rises from the mantle and creates new seafloor. They expected to find volcanic rock, sediment, perhaps a few scattered animals surviving on marine snow. What they found instead was astonishing. Rising from cracks in the seafloor were plumes of water, dark water, clouded with minerals, heated to temperatures above 300 degrees Celsius, hot enough to boil if not for the immense pressure at depth. These vents were surrounded by life. Not sparse life, not scattered animals eking out existence, but dense clusters of organisms, thousands of them. Tube worms taller than a human, clams the size of dinner plates, mussels packed together in mats, crabs crawling over the rocks, fish hovering nearby, all of them thriving, all of them far removed from any connection to sunlight. The scientists had found hydrothermal vents, places where the Earth's internal heat breaks through the ocean floor. And around those vents, life was flourishing. Not surviving, flourishing. This was the edge of a new understanding. A place where the rules changed, where energy came not from above, but from below. Hydrothermal vents form along tectonic boundaries, places where the Earth's crust is thin, where magma sits close to the surface. Seawater seeps down through cracks in the rock. It travels deep into the crust. As it descends, it heats up. The surrounding rock is hot, hundreds of degrees. The water absorbs that heat. It also dissolves minerals from the rock. Sulfur compounds, iron, manganese, hydrogen sulfide. These chemicals saturate the water. Then pressure and heat force the water back up. It rises through the seafloor and erupts into the cold ocean. The temperature difference is extreme. Inside the vent, water reaches 350 degrees Celsius or higher. The surrounding ocean sits near 2 degrees Celsius. When the hot water meets the cold, minerals precipitate. They solidify. They form clouds of black or white particles, depending on the chemistry. These are called black smokers or white smokers. The particles settle around the vent and build up over time. They create chimneys, structures of mineral deposits that can grow meters tall. The vents continue pumping water through them. Hot, chemical-rich, toxic to most life, but not all life. The water emerging from hydrothermal vents contains hydrogen sulfide. In surface environments, this compound is poisonous. It smells like rotten eggs. It disrupts cellular respiration. It is produced by decay, by bacterial breakdown of organic material. But at hydrothermal vents, it is abundant, dissolved in the hot water, mixed with other compounds, and certain bacteria have evolved to use it, not as a toxin, but as fuel. These bacteria perform chemosynthesis, a process fundamentally different from photosynthesis. In photosynthesis, organisms use light energy to convert carbon dioxide and water into glucose. In chemosynthesis, organisms use chemical energy to do the same thing. They do not need light. They need chemicals. Specifically, they need reduced compounds, molecules with electrons available to be stripped away. Hydrogen sulfide is one such compound. The bacteria that live at hydrothermal vents oxidize hydrogen sulfide. They remove electrons from the sulfur atoms. This releases energy. The bacteria capture that energy and use it to build glucose from carbon dioxide and water. The same product as photosynthesis. But the energy source is different. Not light, but chemistry. The reaction is simple in concept, complex in execution. The bacteria must manage extreme heat, extreme pressure, toxic concentrations of chemicals. They cluster around the vents, forming mats on the rocks, coatings on the vent chimneys, floating in the plumes. They multiply rapidly when conditions are right, and they produce organic molecules, sugars, lipids, proteins, the building blocks of life, not from sunlight, but from the Earth's heat. From chemistry alone, these bacteria are the foundation of a new food web. Just as phytoplankton form the base of the surface ecosystem, chemosynthetic bacteria form the base here. Other organisms consume them. Tube worms, clams, mussels, but not by eating them directly in most cases. Instead, through symbiosis. The tube worms found at hydrothermal vents are called Rifschapecaptilla. They grow in dense clusters around the vents. Their bodies are encased in white tubes made of chitin, tubes that can reach over 2 meters in length. At the top of each tube, a bright red plume extends into the water. This plume is not for feeding in the usual sense. It is for gas exchange. The plume absorbs hydrogen sulfide from the vent water. It also absorbs oxygen from the surrounding seawater. Both chemicals are transported into the worm's body. Inside the worm, in a specialized organ called the trophosome, billions of chemosynthetic bacteria live. They are not parasites. They are partners. The worm provides them with hydrogen sulfide and oxygen. The bacteria use those chemicals to perform chemosynthesis. They produce glucose. The worm absorbs the glucose. It uses the sugar for energy, for growth, for reproduction. The worm has no mouth, no digestive system. It does not eat. It is fed entirely by its internal bacteria. This is not rare. Most of the large animals at hydrothermal vents depend on chemosynthetic bacteria. Clams house bacteria in their gills. Mussels do the same. Shrimp have bacteria growing on their backs. Even some snails carry bacteria in specialized organs. The animals and bacteria exist in partnership. Neither can survive without the other. The ecosystem around a hydrothermal vent is dense. Biomass here can exceed that of tropical rainforests. Hundreds of kilograms of living tissue per square meter. All of it sustained by chemistry, by the heat rising from the Earth's mantle, by hydrogen sulfide and other reduced compounds. No sunlight involved. No photosynthesis. The energy comes from tectonics, from the slow churning of the planet's interior, from processes that have nothing to do with the sun. This is fundamentally different from the rest of the ocean. The rest of the ocean depends on the surface, on light penetrating the water, on phytoplankton capturing that light, on animals consuming those phytoplankton. Here, none of that matters. The vent communities are isolated, self-contained, independent. They could exist in total darkness forever. And they do. Hydrothermal vents are not rare. They exist along the entire mid-ocean ridge system, an underwater mountain range that spans more than 60,000 kilometers. It circles the globe like a seam, running down the center of the Atlantic Ocean, curving through the Indian Ocean, cutting across the Pacific. Everywhere tectonic plates pull apart, vents can form, not continuously, but scattered along the ridge, clustered in areas where volcanic activity is strongest. Hundreds of vent fields have been discovered. Probably thousands more exist undiscovered. The ocean is vast. Most of it has never been directly observed. Submersibles can only visit small areas. Remote vehicles scan limited regions. But wherever scientists have looked along active tectonic boundaries, they have found vents. And around those vents, life. Each vent field is unique. The chemistry of the water varies, depending on the underlying rock. Some vents emit water rich in hydrogen sulfide. Others release more methane. Some have high concentrations of iron. Others have copper or zinc. The temperature varies as well. Some vents run extremely hot. Others are cooler, diffuse, seeping rather than erupting. The type of vent determines which bacteria can thrive. Different species specialize in different chemicals. Sulfur-oxidizing bacteria dominate at high sulfide vents. Methane-oxidizing bacteria appear at methane seeps. Iron-oxidizing bacteria coat vents with rust-colored mats. Each bacterial community supports a different set of animals. Tube worms at sulfide vents. Mussels at methane seeps. Yeti crabs at some Pacific vents. The species vary, but the principle remains the same. Life powered by chemistry, independent of light. Vents do not last forever. The mid-ocean ridge is constantly shifting. As tectonic plates move, old vents shut off. New ones open. A single vent might remain active for decades, or centuries, or only years. When a vent stops flowing, the ecosystem collapses. The bacteria die without their chemical fuel. The animals that depend on the bacteria starve. Within months, the dense community disappears. What remains is rock, mineral deposits, empty tubes, shells, a ghost of the vent that was. But elsewhere along the ridge, new vents open, and life colonizes them quickly. Larvae drift in the deep ocean currents. They travel from vent to vent, distances of hundreds of kilometers. When they encounter a new vent, they settle. They mature. They reproduce. The cycle continues. Vent ecosystems are temporary, but the system itself is permanent. As long as tectonics continue, vents will form, and life will find them. Not all chemosynthetic ecosystems depend on hydrothermal vents. Some form at cold seeps, places where methane or hydrogen sulfide seeps up through seafloor sediments. No heat involved, no volcanic activity, just slow release of chemicals from deeper layers of rock. Cold seeps exist on continental margins, areas where sediment is thick, where organic material buried millions of years ago breaks down and releases gas. Methane rises through the sediment. Bacteria in the mud consume it. They oxidize the methane using sulfate from seawater. This reaction produces hydrogen sulfide. Other bacteria oxidize the hydrogen sulfide. Both groups perform chemosynthesis. Both produce organic molecules. And animals cluster around these seeps just as they do at vents. Mussels, clams, tube worms of different species, all hosting symbiotic bacteria, all thriving without sunlight. Cold seeps are even more widespread than hydrothermal vents. They exist on nearly every continental slope, wherever organic-rich sediment accumulates, wherever methane can rise. They are slower than vents, cooler, less dramatic, but just as independent, just as self-sustaining. When hydrothermal vents were first discovered, they seemed exceptional, strange anomalies in an otherwise uniform deep ocean, special places where unusual chemistry allowed life to exist. But as exploration continued, that view shifted. Chemosynthetic ecosystems are not rare exceptions. They are widespread, common, foundational to deep ocean ecology in ways that were not initially understood. Consider the abyssal plain, the flat, muddy seafloor that covers most of the deep ocean. It seems barren compared to hydrothermal vents. No dense clusters of tube worms, no clouds of bacteria, just sediment and the occasional slow-moving animal. But beneath that sediment, chemistry is active. Bacteria live in the mud, millions of them per cubic centimeter. They do not oxidize hydrogen sulfide from vents. They oxidize other compounds: methane, ammonia, iron, manganese, hydrogen gas, any reduced chemical available in the sediment. These bacteria are chemosynthetic. They produce organic molecules just like vent bacteria. But they do it slowly, at a fraction of the rate. They are not visible. They do not form mats. They exist as individual cells scattered through the mud. But they are numerous, and their collective activity is significant. The animals on the abyssal plain eat marine snow, detritus from the surface, but they also consume sediment. And in that sediment are chemosynthetic bacteria. The bacteria provide nutrients, small amounts, but enough to matter. Studies of abyssal food webs show that chemosynthesis contributes to the diet of many deep-sea animals, not as the primary source, but as a supplement, a background input that adds to the energy available. The system is not purely dependent on sunlight after all. It is mixed. Photosynthesis from above, chemosynthesis from below, both contributing, both feeding the ecosystem. The deep ocean is not a single system. It is layered, interwoven, more complex than it first appears. Whale falls represent another form of chemosynthetic habitat. When a whale dies at the surface, its body sinks, slowly at first as gases keep it buoyant, then faster as decomposition proceeds. Eventually, it reaches the seafloor, a massive package of organic material delivered to the deep. Scavengers arrive quickly. Hagfish, sleeper sharks, amphipods. They consume the soft tissue, flesh, blubber, organs. This feeding frenzy can last months. When the soft tissue is gone, the bones remain. And those bones are rich in lipids, oil stored in the bone matrix. Bacteria colonize the bones. They break down the lipids. This process releases hydrogen sulfide, the same chemical found at hydrothermal vents. And the same types of bacteria appear. Chemosynthetic bacteria that oxidize the sulfide. They coat the bones in white mats. Animals that live at vents also appear at whale falls. Certain species of mussels, clams, worms, they settle on the bones and feed on the bacteria. A whale fall can support a chemosynthetic community for decades. 50 years or more for a large whale. During that time, it functions as an island, a patch of high productivity in the deep, sustained by chemistry, by the breakdown of lipids and the production of sulfide. No connection to sunlight except the whale itself. And once the whale is gone, the energy it provided came from feeding at the surface long ago. The chemosynthesis happens in the deep, independent, self-contained for its duration. Wood falls work similarly. Large pieces of driftwood that sink to the seafloor, or logs washed out to sea during storms. Trees do not naturally belong in the ocean, but they arrive. And when they do, they bring cellulose, lignin, complex carbohydrates that few animals can digest, but certain bacteria can. They break down the wood. The process releases hydrogen sulfide. Chemosynthetic bacteria appear. Animals that host those bacteria colonize the wood. Specialized clams, mussels, worms. The wood becomes a temporary ecosystem, lasting years or decades depending on size. Then depleted, consumed, returned to sediment. But while it lasts, it functions without sunlight. Another pocket of chemosynthetic life, hidden on the deep seafloor. The pattern repeats. Anywhere chemicals accumulate, bacteria find them. Anywhere bacteria perform chemosynthesis, animals follow. The deep ocean is scattered with these sites. Vents, seeps, whale falls, wood falls, sunken plant material, pockets of methane, each one a small ecosystem, each one independent. Together they form a network, not connected physically, but connected through dispersal. Larvae drift from site to site. They travel on deep currents. When they find a new chemical source, they settle. The species at vents are often the same species at seeps or at whale falls. They are specialists in chemosynthetic habitats, moving between them, colonizing new ones as old ones fade. This is not rare life. This is a parallel system, operating throughout the deep ocean, invisible from the surface, unknown until recently, but vast. The bacteria that perform chemosynthesis are ancient, far older than photosynthetic organisms. The earliest life on Earth likely used chemosynthesis, not photosynthesis. This seems counterintuitive. Sunlight is abundant, available, constant. Why would life begin with chemistry instead of light? Because early Earth had no oxygen. The atmosphere was mostly nitrogen, carbon dioxide, and water vapor. No ozone layer existed. Ultraviolet radiation from the sun reached the surface unfiltered, lethal to organic molecules, lethal to any cell exposed to it. The surface was hostile, but the deep ocean was not. Protected by water, shielded from radiation, and filled with chemicals. Volcanic vents were likely more numerous then. The Earth's crust was thinner, hotter, more active. Vents released hydrogen, hydrogen sulfide, methane, and other reduced compounds. The same chemicals modern chemosynthetic bacteria use. Early life did not need to invent chemosynthesis. It began with chemosynthesis. It had no other option. Photosynthesis came later, evolved by cyanobacteria perhaps 3 billion years ago. Maybe earlier. The timing is uncertain, but the impact is clear. Photosynthesis produced oxygen. Oxygen accumulated in the atmosphere. It created the ozone layer. It made the surface habitable. It allowed complex life to evolve. The entire history of visible life on Earth depends on photosynthesis. Every plant, every animal, every ecosystem you can see. But chemosynthesis never disappeared. It continued in the deep, unchanged, operating on the same principles as the earliest life, using the same chemicals, the same reactions. The deep ocean preserved a process that predates sunlight-based life, not as a remnant, but as a functioning system. This raises questions about energy availability. Photosynthesis is efficient in its own way. Light is abundant at the surface, free, constant during daylight hours. Plants and phytoplankton can capture it endlessly. They produce massive amounts of biomass. Forests, grasslands, algae blooms. The productivity is enormous. Chemosynthesis is different. The chemicals it requires are limited. Hydrogen sulfide at vents is abundant only near the vent itself. Meters away, it dilutes into the ocean. Methane at seeps is available only where the seep is active. The energy sources are localized, patchy, temporary, A vent field might produce high biomass, but only in a small area, a few hundred square meters, surrounded by kilometers of abyssal plain where productivity is minimal. The total energy input from chemosynthesis is far less than from photosynthesis. Globally, averaged across the ocean, photosynthesis dominates, but locally at the sites where chemosynthesis occurs, it dominates completely. It is not competing with photosynthesis. It is replacing it. The animals at hydrothermal vents are adapted to conditions that seem impossible. Water at 350 degrees Celsius, pressure of several hundred atmospheres, toxic concentrations of hydrogen sulfide and heavy metals, no light, no connection to the surface. These conditions would kill most life instantly. But for vent animals, they are normal. Required even. Tube worms cannot survive away from vents. If removed from the hydrogen sulfide-rich water, they die. Their symbiotic bacteria need the sulfide. Without it, they stop producing glucose. The worm starves. It has no other way to feed. This is not adaptation in the sense of tolerance. This is dependence. The worms are locked into this environment. They cannot leave. They cannot survive anywhere else. Their entire biology is built around chemosynthesis, around vents, around conditions that are lethal to most organisms. The same is true for many vent species. Vent mussels, vent clams, vent shrimp. They exist only at vents or seeps. If the vent stops flowing, they die. If they drift away from the vent, they die. They are not generalists. They are extreme specialists. This creates fragility. Each vent field is isolated, separated from other vent fields by kilometers of abyssal plain. The populations at each vent are small, localized. When a vent shuts off, that population is gone. The species persists only if larvae from other vents arrive and colonize new sites. This dispersal is uncertain. Larvae drift in deep currents for weeks or months. Most die before finding a new vent. The distances are vast. The targets are small. And yet the system works. Vents continue to be colonized. Populations continue. The network persists. How this happens is not fully understood. The deep currents are complex, not well mapped. Larvae are difficult to track. The process remains partly mysterious. The relationship between tube worms and their bacteria is not simple. It is not parasitism. It is not predation. It is mutualism. Both organisms benefit. Neither can survive alone. But the mechanics are strange. Tube worms have no mouth, no gut, no digestive system at all. They absorb nutrients directly from their bacteria. The bacteria live inside a specialized organ. They are not free-living. They cannot exist outside the worm. They are inherited, passed from parent to offspring, or acquired from the environment during larval stages. The exact mechanism varies by species. Some worms are born without bacteria. The larvae must acquire them from seawater after settling. Other species inherit bacteria directly. Either way, once established, the bacteria become permanent, integrated into the worm's biology. They divide when the worm grows. They die when the worm dies. They are not separate organisms anymore. Not entirely. They are something in between. This blurs the concept of individual. What is the tube worm? Is it the animal? Or is it the animal plus the bacteria? Biologically, it is both. The worm cells are animal cells. The bacteria are bacterial cells. Two different lineages. Two different genomes. But functionally, they operate as one unit. One organism. The worm provides transport. It extends its plume into the water. It absorbs hydrogen sulfide. It delivers the sulfide to the bacteria. The bacteria provide food. They convert sulfide and carbon dioxide into glucose. They release the glucose into the worm's tissues. Neither part can function without the other. The worm cannot make food. The bacteria cannot access sulfide. Together they thrive. Separately they die. This is not cooperation. This is fusion. A merging of two life forms into one system. Many deep-sea animals have similar relationships. Vent mussels host bacteria in their gills. The gills are enlarged, modified to house dense bacterial populations. The mussels pump water through their gills. The water contains hydrogen sulfide or methane. The bacteria absorb the chemicals. They perform chemosynthesis. The mussels absorb the glucose. The mussels can also filter feed. They can consume plankton or detritus if available. They have a digestive system. Unlike tube worms, they are not entirely dependent on their bacteria. But the bacteria provide the majority of their nutrition, especially in environments where other food is scarce. Vent shrimp have bacteria growing on their backs. In some species, the bacteria form a thick mat, coating the carapace. The shrimp position themselves in the vent flow, exposing the bacteria to hydrogen sulfide. The bacteria grow. The shrimp harvest them. They scrape the bacteria off and consume them. This is farming. The shrimp cultivate bacteria. They maintain the correct conditions. They harvest the product. The bacteria are not internal symbionts. They are external, cultivated, managed, but the principle is the same. Life sustained by chemosynthetic bacteria. Energy derived from chemicals, not from light. The variety of symbiotic arrangements is surprising. Some animals host bacteria in their gills, others in specialized organs. Some carry bacteria on their surfaces. Others consume free-living bacteria from the water. Each arrangement is different. Each has evolved independently. Symbiosis with chemosynthetic bacteria has arisen many times, in different lineages, in different ways. This suggests it is advantageous, strongly advantageous, to the point where it evolves repeatedly. Whenever animals colonize chemosynthetic habitats, the pattern appears. Bacteria provide energy. Animals provide transport and access to chemicals. The partnership forms and thrives. The animals at hydrothermal vents are not related to each other in obvious ways. Tube worms are annelids, segmented worms related to earthworms. Vent mussels are mollusks, related to oysters and clams. Vent shrimp are crustaceans, related to lobsters and crabs. These groups diverged hundreds of millions of years ago, long before vents were colonized. Yet they all ended up at vents, independently, through separate evolutionary paths. This is convergent evolution. Different lineages arriving at similar solutions. symbiosis with chemosynthetic bacteria, tolerance of extreme conditions, dependence on chemical energy. Each group evolved these traits separately, but the traits are similar because the environment demands them. The species at vents are often unique, found nowhere else, endemic to vent habitats. They do not occur on the abyssal plain. They do not occur at the surface. They are specialists, confined to vents. This isolation is genetic as well as geographic. Populations at different vent fields are separated. Gene flow between them is limited. Larvae disperse, but most do not reach other vents. The populations diverge. Over time, they become distinct. Different species, different adaptations, unique to their particular vent field. This creates high diversity. Each vent field has slightly different conditions, different water chemistry, different temperature, different flow rate. The species adapt to those conditions. They specialize further. They become endemic not just to vents in general, but to specific vents. The discovery of vent ecosystems was surprising, but the discovery of how old they are was more surprising. Fossils of vent animals have been found in ancient rocks, over 300 million years old, preserved in sediments that were once seafloor. The fossils show tube worms, clams, animals that clearly lived at hydrothermal vents. This means vent ecosystems have existed for at least 300 million years, probably longer, possibly since the early ocean formed. These are not new ecosystems. They are ancient, stable, persistent across geological time. They survived mass extinction that wiped out most surface life, the asteroid impact that killed the dinosaurs, the volcanic eruptions that caused other extinctions. These events devastated surface ecosystems, but the deep ocean was largely unaffected. Vents continued flowing. Bacteria continued synthesizing. Animals continued thriving. In darkness, unchanged. This stability is strange. Most ecosystems are dynamic. They change with climate, with sea level, with temperature, but vent ecosystems are buffered. The deep ocean does not experience seasons. It does not experience day and night. Temperature is constant. Pressure is constant. The only changes are local. When a vent stops, when a new one opens, but the system as a whole remains stable. The conditions that support chemosynthesis have not changed in hundreds of millions of years. The Earth's interior is still hot. Tectonics are still active. Chemicals still emerge from the crust. The fuel for chemosynthesis is still available. As long as that remains true, the ecosystems will persist. They are not dependent on external conditions, not dependent on atmospheric composition, not dependent on sunlight intensity. They are insulated, self-contained, stable in a way surface ecosystems are not. Methane is simpler than hydrogen sulfide. One carbon atom bonded to 4 hydrogen atoms. It is produced by decomposition of organic matter. In sediments, in swamps, in the guts of animals. It is a byproduct of anaerobic metabolism. When bacteria break down organic material without oxygen, they release methane. In the deep ocean, methane accumulates in sediments. Organic material from the surface sinks and is buried. Over time, it decomposes. The methane produced rises through the sediment. Sometimes it escapes into the water. Sometimes it gets trapped. Under high pressure and low temperature, methane can form a solid. Methane hydrate, also called methane ice. It looks like ice, feels like ice, but it is not frozen water. It is methane molecules trapped in a cage of water molecules, stable only under specific conditions. High pressure, low temperature, both are present in the deep ocean. Methane hydrate deposits are vast. They exist on continental margins worldwide, buried in sediments, concentrated in certain regions. Estimates suggest there is more carbon stored in methane hydrates than in all fossil fuel reserves combined. Coal, oil, natural gas, all of it combined, and methane hydrates exceed that amount. This is not a small reservoir. It is enormous. Most of it lies at depths between 300 and 2,000 meters. On the continental slopes, where sediment is thick, where organic material accumulates, the methane is locked in the sediment, stable, inert, unless conditions change. Methane seeps occur where hydrates destabilize, where pressure decreases, where temperature increases, where tectonic activity fractures the sediment. The hydrate breaks down, releases methane gas. The gas rises through the sediment. Bubbles emerge from the seafloor, or methane dissolves in seawater and seeps out slowly. Either way, it becomes available, and bacteria colonize it. Methanotrophic bacteria, methane-oxidizing bacteria. They consume methane and produce carbon dioxide. The reaction releases energy. The bacteria use that energy to build organic molecules. Chemosynthesis. Different substrate than hydrogen sulfide, but the same principle. Chemical energy converted to biological energy. The animals at methane seeps are often the same species found at hydrothermal vents, or closely related species. Mussels, clams, tube worms. They host bacteria just as vent animals do. But the bacteria are different species, specialized for methane instead of sulfide. The animals provide the same service. They concentrate the bacteria. They deliver the substrate. They absorb the glucose. The symbiosis is the same, just with different bacteria. This flexibility is notable. Animals can partner with different bacterial species depending on the chemical environment. The partnership is not fixed. It is adaptable. Functional. Methane seeps are quieter than hydrothermal vents. No black smokers. No plumes of heated water. Just slow release of gas. Bubbles rising. Or dissolved methane diffusing into the water. The ecosystems are less dense, slower growing, but still productive, still independent of sunlight, still sustained by chemistry alone. They exist at shallower depths than most hydrothermal vents. Some methane seeps occur at depths of only a few hundred meters, close enough that some sunlight reaches the area. But the seep communities do not use it. They do not need it. They function on methane. Even in the presence of light, they ignore it. Chemosynthesis is their foundation. Their only foundation. The discovery of chemosynthetic ecosystems changed assumptions about where life could exist. For most of history, life was believed to require sunlight, either directly or indirectly. No sunlight meant no energy. No energy meant no life. This seemed fundamental, but chemosynthetic ecosystems disproved it. Life does not require sunlight. It requires energy, and energy can come from sources other than light. Chemistry, specifically redox chemistry, the transfer of electrons between molecules. Any environment with chemical disequilibrium can support life. If reduced compounds and oxidized compounds exist in the same place, if they do not naturally react, if they need a catalyst, then bacteria can provide that catalyst and extract energy in the process. This is a much broader requirement than sunlight. On Earth, redox disequilibrium exists at hydrothermal vents, at methane seeps, in deep sediments, in certain caves, in certain underground aquifers, anywhere volcanic or tectonic activity releases reduced chemicals, anywhere decomposition produces methane or hydrogen sulfide. These environments are not rare. They are common, just hidden. But beyond Earth, similar environments could exist. Mars has subsurface water, possibly liquid. It has volcanic history. If chemical energy is available, life could exist. Europa, a moon of Jupiter, has an ocean beneath its ice shell. The ocean may be in contact with a rocky mantle. Hydrothermal vents could exist there. Chemosynthetic ecosystems could exist there. Without any sunlight reaching the surface, without any photosynthesis, life sustained by the moon's internal heat, by chemistry alone. This is speculative. No life has been found on Europa, or Mars, or anywhere beyond Earth. But the possibility exists, and it is not science fiction. It is based on known processes, on observed ecosystems, on chemistry that works the same way everywhere. The requirements for chemosynthesis are simpler than for photosynthesis. No light needed. No oxygen needed initially. Just water, carbon dioxide, reduced chemicals, heat. All of these exist in many places in the solar system. They likely exist on moons of Saturn, on planets around other stars, anywhere geological activity persists, anywhere water exists. Life might exist, not dependent on sunlight, not dependent on a star, independent, hidden, operating on principles discovered only recently on Earth. Bacteria divide quickly under optimal conditions. At hydrothermal vents, some species can double their population in hours. The sulfide is abundant. The temperature is tolerable. The energy flows continuously. They coat the rocks in thick mats, white, yellow, orange depending on the species and the minerals present. The mat grows until it reaches equilibrium, until the rate of growth matches the rate of consumption. Animals graze on the bacteria. Shrimp scrape them from surfaces. Snails rasp them with specialized tongues. Crabs pick through them with delicate claws. The mat regenerates constantly, a living surface, renewable. Tube worms grow differently. They grow slowly. A large Rifshapachyptilla may be 40 years old or older. The tubes record their growth in layers, like tree rings, but the process is gradual, a few centimeters per year. sometimes less. Growth depends on the vent, on the flow of sulfide. When the flow is strong, the worm grows faster. When it weakens, growth slows, stops entirely in some cases. The worm waits, conserving energy, surviving on stored reserves. Months can pass this way, years even, until the vent flow resumes, or until it stops completely, and the worm dies. Clams at cold seeps live even longer. Centuries. A single clam can be 500 years old, measured by isotope dating, by growth rings in the shell. These clams grow at rates measured in millimeters per decade, almost imperceptible. Their metabolism is extremely slow. Their bacteria produce glucose continuously, but at very low rates. The seep provides methane steadily, but not abundantly. The clam does not need rapid growth. It needs persistence. Time replaces speed. Longevity replaces productivity. The clam outlasts changes in the environment, outlasts variations in methane flow, outlasts predators. It sits in the sediment, half buried, filtering water, feeding its bacteria, absorbing glucose, growing so slowly that movement is invisible. In the surface ocean, most fish live a few years, a decade at most. They grow quickly, reproduce annually, die young. In the deep ocean, fish live longer, much longer. Orange roughy, a deep-sea fish, can live over 100 years, possibly 200. Growth is slow. Reproduction happens late. After decades, eggs are few. Larvae drift for months. Most die. The survivors take years to mature. This is not inefficiency. This is adaptation. The deep ocean provides little food. Growth requires energy. Energy is scarce. Fast growth is wasteful. Slow growth conserves resources, allows survival during long periods without food. Time is abundant. Why rush? Vent fields appear and disappear on timescales of decades. Individual vents within a field can last years or centuries. Cold seeps persist for thousands of years, tens of thousands in some cases. The ecosystems that depend on them shift accordingly. Fast turnover at young vents, slow persistence at old seeps. The animals match their life history to the habitat duration. Vent tube worms live decades, long enough to reproduce at one vent, but not so long that they outlive the vent itself. Seep clams live centuries, matching the longevity of the seep, surviving fluctuations, waiting through dormant periods, persisting until conditions improve. At the surface, atmospheric pressure is 1 bar. The weight of the air above, 14.7 pounds per square inch. This is the pressure your body is built for. In water, pressure increases with depth. Every 10 meters adds approximately 1 bar. At 1,000 meters, pressure is 100 bars. At 4,000 meters, 400 bars. At the deepest trenches, over 1,000 bars. This is not a subtle increase. This is crushing force, enough to collapse air-filled spaces instantly. Human lungs, submarine hulls, and less specifically reinforced. Anything with air inside compresses, fails, implodes. Deep-sea organisms do not have air spaces. No lungs, no swim bladders, Their bodies are filled with water. Incompressible. The pressure outside matches the pressure inside. No crushing force. But pressure still affects biology. It changes how molecules interact. Proteins fold differently under high pressure. Membranes become rigid. Chemical reactions slow down. Enzymes that work at surface pressure fail at depth. Deep-sea organisms compensate. Their proteins are modified. Different amino acid sequences. Different shapes. Structures that remain flexible under pressure. Their membranes contain different lipids. More unsaturated fats. Remaining fluid despite the pressure. Their enzymes are adapted. Functioning optimally at 400 bars. Failing at surface pressure. This creates a barrier. Deep-sea animals brought to the surface die. Not from decompression. Their bodies do not rupture. But their biochemistry fails. Proteins unfold. Membranes become too fluid. Enzymes stop working correctly. Metabolism collapses. Similarly, surface animals cannot survive at depth. Their proteins collapse. Their enzymes fail. Their biochemistry is pressure sensitive. Optimized for 1 bar. Fatal at 400 bars. The depth barrier is absolute. Not physical. Biochemical. Some fish migrate vertically. From deep water to shallower water. They cross pressure gradients of hundreds of bars. Somehow their biochemistry tolerates this. They have flexible proteins. Enzymes that function across a range of pressures. But this flexibility comes at a cost. They are not optimized for any single depth. They perform adequately at many depths, but not excellently at any. True deep-sea specialists do not migrate. They remain at depth. Fixed. Adapted so specifically that they cannot leave. Vision requires light. Eyes evolve to detect light, to distinguish shapes, to track movement, to identify color. At the surface, vision is universal. Nearly every animal has eyes. Some simple, some complex, but present. In the deep ocean, eyes become less common, or they change. In the twilight zone, eyes enlarge. Pupils widen. Retinas pack with more light-sensitive cells. These fish detect the faintest traces of sunlight filtering down. Or they detect bioluminescence, the light produced by other organisms. But below 1,000 meters, sunlight is absent. Completely. Bioluminescence still exists. Some animals produce light to hunt, to communicate, to confuse predators. But many animals have no eyes at all, or reduced eyes. Non-functional. Vision is expensive. Building eyes requires energy. Maintaining them requires energy. Processing visual information requires brain tissue. If there is nothing to see, these costs are wasted. Evolution removes waste. Eyes shrink, disappear, replaced by other senses. Chemoreception becomes primary, detecting dissolved chemicals in water, amino acids, proteins, molecules released by prey, by predators, by potential mates. The ocean is full of chemical traces, invisible to eyes, but detectable by receptors. Animals at depth have elaborate chemosensory organs. Long antennae, barbels, specialized skin cells. They taste the water constantly. building a chemical map of their surroundings. Who is nearby? What they are? Whether they are food. This sense is acute, sensitive to parts per billion, able to track gradients over long distances, following a chemical trail to its source. Hours of slow swimming, guided only by scent. Mechanoreception also dominates, detecting vibrations, pressure waves, movement in water. Fish have lateral line systems, rows of sensors along their bodies. These detect disturbances, a struggling prey, a passing predator, water currents shifting. In darkness, vibration carries information that light cannot. It travels farther, persists longer, cannot be hidden by intervening objects the way light can. Animals at depth move carefully, slowly, minimizing vibrations, becoming invisible to mechanoreceptors. Or they move suddenly, rapidly, bursts of motion to cross open water. Then stillness again. Sound travels well in water, better than in air, faster, farther. But the deep ocean is quiet. No wind, no waves, no rain, no ice. The sounds that fill shallow water do not reach here. What remains is subtle. The click of shrimp, the low-frequency calls of whales passing through, tectonic groans from the seafloor shifting, but mostly silence. Animals here are quiet. They do not vocalize. They do not need to. Communication happens through chemicals, through bioluminescence if they have it, through touch. Sound is not useful. It fades into the background, ignored. Bioluminescence is common but not universal. Many deep-sea fish produce light through symbiotic bacteria or through chemical reactions in specialized cells called photophores. They use light to lure prey. Anglerfish dangle glowing lures in front of their mouths. Prey approaches, drawn to the light. The anglerfish strikes. Some fish use light defensively. They emit sudden bright flashes, blinding predators or confusing them, allowing escape. Some use bioluminescence for camouflage. They produce light on their undersides, matching the faint glow from above, erasing their silhouette, becoming invisible from below. This is called counterillumination. It works in the twilight zone, where faint sunlight still penetrates. Deeper, it is irrelevant. No light from above to match. At hydrothermal vents, animals do not produce light. Or they do, but it serves no purpose. The environment is too bright in its own way. Not with visible light, but with heat, with infrared radiation. Some vent shrimp have modified eyes, not for seeing visible light, for detecting infrared heat. They use these sensors to navigate near vents, staying close enough to feed, but not so close they cook. The distinction is narrow. Centimeters, a few degrees of temperature difference. The shrimp sense the gradient, position themselves precisely. Most vent animals have no eyes. They do not need them. The vent is a chemical beacon, a plume of sulfide rising into the water, detectable by chemoreceptors from hundreds of meters away. Following the plume leads to the source, to the vent, to the food. Vision would add nothing. Darkness is not an obstacle here. It is the default condition. Expected. Adapted to so completely that light becomes irrelevant. Even if light were present, it would not be used. The sensory systems are built for chemistry, for heat gradients, for vibrations, for a world where darkness is normal, where it has always been normal, where no other condition exists. Water at a vent does not simply rise. It pulses, variable flow depending on subsurface conditions, on pressure in the crust, on the pathways through rock, sometimes strong, a jet of superheated water forcing its way through a narrow opening, sometimes weak, diffuse seepage through cracks. The animals respond to these variations. Tube worms extend their plumes when flow is strong. Sulfide is abundant. The bacteria produce glucose rapidly. The worm grows. When flow weakens, plumes retract. Tucked into the tube. Protected. The worm waits. Metabolism slows. Stored reserves are consumed. Days pass. Weeks. Flow resumes or it does not. If it does, the cycle continues. If it does not, the worm eventually dies. There is no decision involved. No behavior. Just response to chemical availability. Presence or absence. Muscles packed together around seeps. Beds of them. Shells touching. Overlapping. Competing for space. For position closest to the methane source. Those on the periphery receive less methane. Their bacteria are less productive. The muscles grow more slowly. Reproduce less. Those at the center dominate. They anchor to rock, to each other, layer upon layer. New muscles settle on old ones. The bed builds upward. Older muscles get buried, covered by younger individuals. They lose access to the methane. Their bacteria starve. The muscle dies. Its shell remains. Foundation for the next layer. This is not predation, not competition in the active sense, just accumulation, a consequence of limited space and abundant larvae. Crabs move through the beds, picking at dead mussels, consuming soft tissue. They do not hunt the living mussels. The shells are too strong, but dead ones open, exposed. The crabs are scavengers here, not predators. They wait for death, for shells to gap open, for access. Patient. There is always something dead. Turnover is constant, not rapid, but steady. A mussel dies. A crab finds it within hours, within a day. Consumption takes time. The crab feeds slowly. Other crabs may join. They do not fight. They share. There is enough. More mussels will die. More will always die. The seep persists. Methane continues seeping through sediment, rising through cracks, dissolving into water. Bubbles form sometimes. Visible streams of gas rising from the mud. They ascend a few meters, then dissolve. The pressure keeps them from reaching the surface. They hydrate, become methane hydrate crystals, suspended in the water column, eventually settling back to the seafloor. The cycle repeats. Methane locked in sediment, released slowly, consumed by bacteria. Partially, some escapes into the water, rises, hydrates, sinks, gets incorporated into sediment again. Buried, released again decades or centuries later. Circulating, temperature at depth is stable, 2 degrees Celsius. Year-round, no seasons, no daily variation, just constant cold. Water this cold is dense, heavy, it sinks, displaces warmer water, drives ocean circulation. But the animals do not experience circulation. They experience stillness. The large-scale currents move slowly, centimeters per second, imperceptible. Water feels stationary, stagnant even, but it is not. It moves, carries dissolved oxygen, nutrients, larvae, chemical signals. Everything drifts with it, suspended, transported over distances impossible to detect locally. Metabolism slows in cold water. Chemical reactions proceed at reduced rates. Enzymes work more slowly. Diffusion is slower. Oxygen uptake is slower. Waste removal is slower. Everything about biology becomes sluggish. An animal at 2 degrees operates at a fraction of the speed of the same animal at 20 degrees. Growth is slower. Healing is slower. Digestion is slower. Reproduction is slower. But energy demands are also lower. Less heat is lost. No energy spent maintaining body temperature. The cold is not fought. It is accepted, incorporated. Animals here are ectothermic. Their body temperature matches the water. Cold-blooded, though the term is imprecise. Their blood is not cold by choice. It is cold because the environment is cold, and their biology is built for it. Hydrothermal vents disrupt this. Water at the vent opening can exceed 300 degrees. Hot enough to denature proteins instantly. Hot enough to kill. But a few centimeters away, temperature drops, mixing with surrounding water, cooling rapidly. At 10 centimeters from the vent, water might be 50 degrees. At 1 meter, 10 degrees. At 10 meters, 3 degrees. Back to ambient. The gradient is steep. Precise positioning matters. Tube worms grow right at the boundary, where hot vent water mixes with cold ocean water, where temperature is tolerable, 20 to 30 degrees perhaps. Warm for the deep ocean, but not lethal. Their plumes extend into the mixed zone, absorbing sulfide from the hot water, absorbing oxygen from the cold water. Both are needed, both delivered by the precise location. Too close to the vent, and the worm overheats. Tissues cook. Proteins denature. Death is rapid. Too far from the vent, and sulfide concentration drops. Bacteria become less productive. The worm starves slowly. The optimal zone is narrow. The worm cannot move, cannot adjust position once settled. Its location is fixed, permanent. Either conditions remain favorable or they do not. Vent shrimp cluster at the edges, where temperature is highest but still survivable. 40 degrees, 50. They tolerate heat that would kill most deep-sea animals. Their proteins are heat-stable, modified, adapted, but they still have limits. They position carefully, moving away if water gets too hot, moving closer if it cools. Constant adjustment, reading the gradient, staying within the viable range. When vents pulse stronger, they retreat. When flow weakens, they advance, always seeking the optimal temperature, the optimal sulfide concentration, the place where their bacteria grow fastest. Some vent animals tolerate extraordinary heat. Pompeii worms live inside vent chimneys, tubes built into the mineral structure. The water flowing past them can reach 80 degrees. The hottest temperature any animal is known to survive continuously. Their bodies are coated in bacteria. The bacteria form an insulating layer. They also consume sulfide, providing food. The worm is both protected and fed by its bacterial coating. Without the bacteria, the worm would overheat, or starve, or both. The partnership is obligate. The worm cannot exist without the bacteria. The bacteria cannot exist without the worm providing substrate and position. Together they inhabit conditions nothing else can tolerate. The hottest biological environment on Earth. Not at the surface, but 2,000 meters below it. In darkness, in a place that should be cold, but is not, because the Earth's interior heat breaks through, and life meets it there. Reproduction at vents is not synchronized. No seasons exist to cue spawning. No temperature change. No light cycle. Animals reproduce when conditions permit, when energy is sufficient, when the vent is flowing strongly, when the animal has grown enough. Timing varies. Some tube worms release eggs continuously. Small numbers, trickling into the water over months. Others spawn in pulses, irregular intervals, responding to internal cues, or to environmental fluctuations too subtle to measure. Eggs drift away immediately, carried by currents. Fertilization happens in the water column. Males release sperm. Females release eggs. They do not pair, do not coordinate. Gametes meet by chance, diluted in seawater. Most do not fertilize. Most eggs drift unfertilized. Die, sink, get consumed by filter feeders. Larvae that develop drift for weeks. They are planktonic, floating, unable to swim effectively against currents. They have no symbiotic bacteria yet. They feed on yolk reserves, or on dissolved organic matter, or on bacteria in the water. But the feeding is minimal. They are not growing, just surviving, drifting, searching passively for a new vent. Most never find one. They drift into regions without vents, starve, sink to the abyssal plain, die there. Their bodies join the marine snow, feed the animals of the plain, transfer a tiny amount of vent-derived energy into the wider deep ocean. Larvae that happen to drift near a vent detect it. Chemical sensors trigger settlement. The larva swims downward, toward the source, toward the sulfide plume. It settles on rock near the vent, cements itself, begins building a tube if it is a tube worm, or burrows if it is a clam, or attaches if it is a mussel. The juvenile is now sessile, fixed in place. It begins acquiring bacteria. Some species inherit bacteria from the parent. Eggs contain bacterial cells already. Others acquire bacteria from the environment. Free-living bacteria invent water. They enter through the skin, through the mouth or gills if present. They colonize internal tissues, multiply, establish the symbiosis. This process takes days, weeks. During that time, the juvenile is vulnerable. It has no food source yet, no glucose production. It survives on stored reserves. If bacteria do not colonize successfully, the juvenile dies. If they do, it begins to grow. Slowly. Predation is limited. Few predators specialize on vent animals. The densest populations are tube worms, clams, mussels, all protected by tubes or shells. Hard structures, difficult to breach. Crabs feed on them, but only when they die. Fish hover near vents. They eat smaller prey. Amphipods, copepods, small crustaceans that graze on bacterial mats. But the fish do not eat the large sessile animals. Cannot. The defenses are too strong. Predation pressure is low. Death comes from other causes. Vents shut down. Burial under new mineral deposits. Overgrowth by neighboring animals. Competition for space. For position. For access to sulfide. Space is the primary limit. Near vents, suitable substrate is rare. Most of the seafloor is sediment. Soft. Unstable. Sessile animals need hard surfaces. Rock. Old vent chimneys. The shells of dead animals. Competition for these surfaces is intense. Larvae settle in dense aggregations. Thousands per square meter. Most do not survive to adulthood. They are overgrown, outcompeted, smothered by neighbors. Only a few individuals per cohort reach maturity. Those few reproduce. The cycle continues. High larval mortality. High reproductive output. Balancing. Most vent animals are sessile. They do not move once settled. Their entire adult life is spent in one location, centimeters from where they first attached. This seems limiting, but movement is not necessary. The vent delivers everything needed. Sulfide, oxygen, dissolved nutrients. Mates are nearby. Spawning releases gametes into the same water. Fertilization happens. There is no benefit to moving. And significant cost. Energy spent on locomotion. Risk of leaving the vent environment. Risk of failing to find a better location. Sessility is efficient. Fixed. Simple. Mobile animals exist at vents. Crabs, shrimp, fish, snails. They move between vents. Not frequently, but occasionally. When a vent weakens, they leave. Travel across the abyssal plain, following chemical gradients, seeking another vent. The journey is risky. No food on the plain. No sulfide. No bacterial mats to graze. They survive on stored reserves. Body fat. Protein reserves. They can travel for weeks. Months perhaps. Moving slowly. Conserving energy. Following faint traces of sulfide dissolved in the water. Following currents that carry vent plumes. Some find new vents. Settle there. Resume feeding. Survive. Others do not. They wander until reserves are exhausted. Stop. Sink to the seafloor. Die. Their bodies are scavenged. Consumed by abyssal scavengers. Amphipods. Isopods. Vish. The energy that originated at one vent gets distributed into the wider ecosystem. A small transfer, but multiplied across many individuals, across many vents. The deep ocean is not completely isolated from vent systems. There is leakage, movement, connection through the animals that travel, through the larvae that drift, through the organic matter that is exported. Vent fish are not specialized feeders. They eat whatever is available. Small crustaceans at vents, amphipods on the abyssal plain, dead animals anywhere. They are opportunists, generalists. This allows them to survive between vents, to travel, to connect populations. But they do not host bacteria. They do not perform chemosynthesis. They are dependent on the vent community, on the animals that do host bacteria, on the productivity those animals create. Without the sessile animals, the fish would have no reason to visit vents. No food source. The sessile animals are the foundation. The fish are secondary, dependent, connected but not integral. Currents at depth are weak but directional. They follow the contours of the seafloor, channeled by ridges and valleys. Animals drifting in these currents move predictably, not randomly. Larvae released at one vent are more likely to reach certain downstream vents, less likely to reach upstream ones. This creates patterns. Gene flow is directional. Populations connected in one direction, isolated in the reverse. Over time, genetic differences accumulate. Populations diverge, become distinct, but they remain connected enough to be the same species, just shaped by geography, by the orientation of currents, by the positions of vents along the ridge. Away from vents, the seafloor is soft. Sediment accumulated over millions of years. Fine particles, clay, silt, organic debris. It forms a layer meters thick, tens of meters in some places. Animals live in this sediment, not on it, in it. Worms burrow through it, creating tunnels, networks of tubes. Water flows through these tubes, bringing oxygen, removing waste. The worms filter the sediment, digest organic particles, extract what little nutrition exists. The rest is excreted, passed back into the sediment, compacted, reworked. Bacteria dominate sediment, outnumber animals by orders of magnitude, trillions of cells per cubic meter. They coat every particle, fill every pore space, metabolize whatever substrates are available. Oxygen in the top layer, nitrate below that, sulfate deeper, methane deeper still. Each layer has different bacterial communities, specialized for the available chemistry. The animals consume these bacteria, directly or indirectly. They irrigate the sediment, bring fresh seawater into deeper layers. The bacteria respond, grow more actively. The animals benefit. More bacteria to consume. Symbiosis without integration. Engineering without intent. Some sediment bacteria are chemosynthetic. They oxidize methane seeping up from below, or hydrogen sulfide produced by decay, or ammonia released by animal waste. They produce organic molecules, small amounts, distributed through the sediment. This production is invisible, unmeasurable without careful chemical analysis. But it exists. Background chemosynthesis. Constant. Widespread. Not concentrated at vents or seeps, but diffuse. Present everywhere chemical gradients exist. Everywhere reduced and oxidized compounds meet. The entire sediment layer is chemosynthetically active, just at very low intensity. In some regions, sediment is thicker. Continental margins, areas where rivers once delivered huge loads of organic material. This material gets buried, compressed. Over millions of years, it alters. Organic molecules break down, release methane. The methane migrates upward, slowly, through pore spaces, through cracks. Some reaches the surface, forms cold seeps. Most remains trapped, bound in hydrates, locked in sediment, unavailable, but present, waiting. If pressure decreases or temperature increases, the hydrate becomes unstable, releases methane. The methane rises rapidly. Seeps intensify. Bacteria respond. Populations bloom. Animals colonize. The seep ecosystem expands, then stabilizes, or declines if methane flux decreases, fluctuating over decades, centuries, responding to conditions deep in the sediment, conditions invisible from the surface. The surface ocean is bright, warm, rich with life. Phytoplankton bloom. Fish school in millions. Whales migrate. Seabirds dive. Energy flows rapidly, constantly, driven by continuous sunlight. The deep ocean is dark, cold, sparse. Animals are scattered, moving slowly, eating rarely. Energy flows slowly, intermittently, driven by rare inputs. These are not the same system. They are layered, occupying the same space vertically, but separated by depth, by temperature, by pressure, by light. Connection exists. Marine snow falls. Animals migrate vertically, but the connection is weak. Most life in each layer never interacts with the other. Chemosynthetic ecosystems are separate even from the deep ocean. They do not depend on marine snow, do not depend on oxygen produced by phytoplankton. They could function if the surface ocean disappeared. If photosynthesis stopped entirely, the vents would continue. The bacteria would continue. The animals would continue. Isolated, independent, operating on energy from the Earth's interior. This is not a thought experiment. It is fact. Measured, observed. Vent ecosystems are fundamentally disconnected from the rest of the biosphere. Not geographically, not evolutionarily, but energetically. Their energy does not come from the sun. It comes from tectonics, from chemical reactions in the crust, from heat rising from the mantle, from processes that have nothing to do with life on the surface. In the past, Earth's surface was different. The atmosphere contained no oxygen. Photosynthesis had not yet evolved, or had just begun. Oxygen levels were negligible. The surface ocean was anoxic, hostile to complex life. But the deep ocean was the same as it is now. Vents existed. Chemosynthetic bacteria existed. Animals had not evolved yet, but the bacteria were there, operating, producing organic molecules, sustaining themselves. When animals did evolve, millions of years later, they found the vents, colonized them, formed the ecosystems that exist today. Those ecosystems are essentially unchanged. The conditions that support them have not changed. The bacteria are the same. The chemistry is the same. The energy source is the same. Time has passed, hundreds of millions of years, but the deep ocean remains stable. The vents remain active. The system persists. If a disaster occurred at the surface, an asteroid impact, a supervolcano, a gamma ray burst, any event that blocked sunlight for years, photosynthesis would stop. Surface ecosystems would collapse. Most life would die. But the deep ocean would be unaffected. Vents would continue flowing. Bacteria would continue synthesizing. Animals at vents would survive, thrive even, unaware of the catastrophe above. This has likely happened before. During mass extinctions, surface life was devastated. Coral reefs died. Forests burned. Phytoplankton populations crashed. But deep-sea life persisted. Vents were not affected. The fossil record shows this. Vent animals exist in rocks from before and after extinction events. The lineages are unbroken, continuous, while surface life was replaced, reinvented, rebuilt from survivors. The deep ocean continued, stable, isolated, indifferent to surface conditions. Vent water is saturated with dissolved metals. Iron, copper, zinc, manganese, cadmium, lead, concentrations far higher than normal seawater. These metals come from the rocks the water passes through, leached out by heat and pressure, carried upward, released at the vent. When the hot metal-rich water meets cold seawater, the metals precipitate, solidify, form particles. These particles build up around the vent, upon layer, creating chimneys, towers of metal sulfides and oxides. Some chimneys grow meters tall, 10 meters, 20. Structures of solid mineral, porous, fragile in structure but massive in volume. The chimneys are not permanent. They collapse eventually, undermined by their own weight, by shifts in vent flow, by tectonic movements. When they fall, they shatter. Fragments scatter across the seafloor. New chimneys build in their place. The cycle continues. The minerals in vent chimneys are economically valuable. High concentrations of copper and zinc, rare earth elements, gold in some cases. Mining companies have investigated these deposits, considered extraction, but the challenges are significant. The depth, the remoteness, the instability of the chimneys, the environmental impact on vent ecosystems. No large-scale mining has occurred yet, but the potential exists. The minerals are there, accessible, concentrated, worth billions potentially. Whether that value outweighs the cost and impact remains debated. Bacteria also interact with metals. Some species oxidize iron. Others oxidize manganese. These reactions release energy. Energy used for chemosynthesis. Iron oxidizing bacteria coat vent chimneys in rust-colored mats. Thick, slimy, filled with precipitated iron oxides. The bacteria are embedded in these mats. Protected, fed by the continuous flow of iron-rich water. Manganese oxidizing bacteria form black coatings. Darker, thinner, equally productive. These bacteria do not form symbioses with animals. They are free-living, grazed by snails, limpets, small crustaceans, but not integrated into tissues, not hosted internally. The relationship is simpler. Producer and consumer, plant and herbivore, but without sunlight, without photosynthesis. The bacteria are primary producers, using metals instead of light. Metal concentrations at vents are toxic to most organisms. Surface animals would die quickly, poisoned by copper, by zinc, by cadmium. But vent animals tolerate these concentrations. Their proteins are modified. Binding sites for metals are altered. Metals are sequestered in specialized cells, stored safely, prevented from interfering with cellular processes. This tolerance is specific. Each species adapted to the particular metal profile of its local vent. Vents in different regions have different chemistry, different metal ratios. The animals reflect this. Populations at copper-rich vents tolerate high copper. Populations at zinc-rich vents tolerate high zinc. Move an animal from one vent to another with different chemistry, and it may not survive. Even though both are vents, even though both have sulfide. The specifics matter. Adaptation is precise, narrow, optimized for local conditions, not generalizable. Between vents, the abyssal plane stretches. Flat, featureless. Sediment covers everything. No rocks protrude. No structure breaks the monotony. Animals here are sparse. A sea cucumber crawling across the mud, moving centimeters per hour, leaving a trail, a furrow in the sediment, marking its passage. The trail remains for weeks, months. Nothing disturbs it. No current strong enough to erase it. Eventually it fills in, slowly, as sediment settles, as other animals cross it. But the process is gradual. Time operates differently here. A fish hovers motionless, waiting for what is unclear, for movement perhaps, for a chemical signal, for prey. Hours pass. The fish does not move, does not need to. Metabolism is slow. Energy requirements are minimal. Waiting costs almost nothing. Eventually something passes. An amphipod, a copepod. The fish strikes. A single rapid movement. Then stillness again. The prey is consumed, digested over days, weeks. One meal sustaining the fish for a long time. No need to hunt frequently. No benefit to constant activity. Stillness is efficient, conserves energy, extends survival between meals. The sediment is not empty. It appears empty, but beneath the surface, activity continues. Worms burrow. Bacteria metabolize. Chemical gradients shift. Pore water circulates. Slowly, driven by the movements of animals, by diffusion, by temperature differences too small to measure. This activity is invisible, requires instruments to detect, but it is continuous. The sediment is alive, just not visibly, not in ways that register to observation. It functions quietly, patiently, processing what settles from above, recycling it, returning nutrients to the water column, feeding bacteria, feeding the animals that feed on bacteria. A system operating at the edge of measurability, but operating nonetheless. Some animals move between environments, between the abyssal plain and hydrothermal vents, between cold seeps and open seafloor. They are not specialists, not entirely. They tolerate the conditions at vents, but do not require them. They benefit from the productivity, but can survive without it. Octopuses visit vents, hunt there, feed on crabs and smaller animals, then leave, return to the plain, to crevices in rock outcrops, to temporary shelters. They are visitors, not residents. The distinction matters. Residents depend on the vent, cannot leave, cannot survive elsewhere. Visitors use the vent as a resource, one resource among others. They are flexible, adaptable, not locked into a single environment. This flexibility comes at a cost. Visitors are not optimized for vent conditions. They tolerate them but do not thrive in them. They compete poorly with specialists, cannot feed as efficiently, cannot grow as rapidly. But they survive when vents shut down. When specialists die, visitors leave, find other food sources, other habitats. They lose the benefit of the vent, but do not lose everything. Specialists lose everything, fixed in place, unable to relocate. When the vent dies, they die. No alternative, no backup. This is the trade-off. Specialization offers high productivity in specific conditions, but zero productivity elsewhere. Flexibility offers lower productivity everywhere, but survival across a range of conditions. Neither strategy is superior. Both exist. Both persist, shaped by different selective pressures, different risks, different environments. The boundary between vent and plain is not sharp. There is a gradient. Near the vent, sulfide concentration is high. Temperature is elevated. Bacterial productivity is high. Animal density is high. Moving away, sulfide decreases. Temperature decreases. Bacterial productivity decreases. Animal density decreases. At some distance, conditions match the surrounding plain. No more sulfide. No more elevated temperature. No more bacterial mats. The transition takes meters, tens of meters. A gradual shift from one ecosystem to another. Animals occupy different positions along this gradient. Tube worms cluster closest to the vent, requiring high sulfide. Mussels spread farther, tolerating lower concentrations. Crabs range widely, feeding at the vent, scavenging on the plain, moving between zones. Each species positioned according to its requirements, its tolerances, its competitive ability. The gradient is structured, organized, not by design, but by the cumulative effect of individual limits. Individual needs. Vent chimneys grow slowly. Millimeters per year. Centimeters in some cases. The minerals precipitate from the water. Settle. Build up. Layer by layer. The structure becomes taller, thicker, more complex. Side vents open. Branch chimneys form. The flow splits, diverges. Each branch continues building, creating elaborate structures, branching towers, networks of channels, all formed by precipitation, by chemistry alone. No biological input, no construction, just deposition, accumulation. The result looks intentional, designed, but it is not. It is the natural consequence of fluid dynamics and mineral chemistry. Water flows through the path of least resistance. Minerals precipitate where temperature and chemistry favor it. The structure grows along those lines. Predictable, repeatable, inevitable given the conditions. Animals colonize chimneys as they grow. Tube worms settle on the sides. Their tubes add to the structure. Not mineral, but organic. Chitin, protein, durable. The tubes remain after the worm dies. Empty, hollow. They become substrate for new settlers. Mussels attach to old tubes. Barnacles, anemones. The chimney becomes layered. Mineral and biological material intermingled. Difficult to separate. When the chimney collapses, both fall together. Shattered fragments, mineral and shell, scattered across the seafloor. Indistinguishable after time. Incorporated into sediment. Buried, preserved, becoming part of the geological record. Future fossils, evidence of the vent that was. Some chimneys are ancient, inactive for decades, centuries, no longer emitting water, no longer supporting chemosynthetic life, but still standing, stable enough to remain upright. Erosion is slow underwater. No wind, no rain, just slow dissolution. Chemical weathering, biological boring, organisms that drill into rock. Sponges, worms, slowly breaking down the structure. But the process takes time. The chimney persists, a monument to past activity. Empty now, silent, but unchanged in form. New vents open near old chimneys sometimes. The new vent reactivates the area. Animals colonize the new vent. The old chimney becomes secondary habitat, not producing sulfide, but providing structure. Hard surface. Larvae settle on it. Animals shelter in it. Use it as substrate. The chimney has a second life. Passive now, but still functional. Still part of the ecosystem. Contributing structure, even without contributing chemistry. A vent opens. How this happens is subsurface. Invisible. Magma rises. Cracks form in the crust. Water seeps down. Heat. Rises. Emerges. The first flow is diffuse. Scattered seepage. Warm water rising through cracks. Not concentrated yet. Bacteria arrive quickly. Within days. Carried by currents. They detect the sulfide. Settle. Begin growing. multiplying, coating the rocks. Larvae arrive later. Weeks, months, drifting from other vents. They settle, metamorphose, begin building tubes, growing shells, establishing themselves. The vent community forms gradually, not instantly, over years. First bacteria, then small animals, then larger ones, then predators, then scavengers. Each arrival depending on the presence of the previous, each step enabling the next. The ecosystem assembles, matures, reaches a stable state. Dense, productive, thriving. This state persists. Decades. The vent flows continuously. Animals grow, reproduce, die, are replaced. The community turns over. Individuals change, but the structure remains. The composition remains. Stable, predictable, a mature vent ecosystem. Then something changes. Subsurface. The flow weakens. Not suddenly, gradually, over months, years. Sulfide concentration decreases. Temperature decreases. The bacteria grow more slowly. Animals grow more slowly. Reproduction decreases. Recruitment decreases. The population ages. Fewer juveniles. More old individuals. Mortality exceeds recruitment. The community shrinks. Eventually the vent stops. No more flow. No more sulfide. The bacteria die within days. Weeks. The animals starve. Tube worms retract. Stop feeding. Consume stored reserves. Last weeks. Months. Then die. Muscles close. Stop filtering. Die more slowly. Months. Years in some cases. Clams persist longest. Metabolism slowed. Barely alive. Years. Decades. But eventually they too die. The vent is silent. Empty. The tubes remain. The shells remain. But the animals are gone. This is normal. Expected. Vents are temporary. Every vent that opens will eventually close. Every community that forms will eventually collapse. The timescale varies. Some vents last centuries. Some last decades. Some only years. But none last forever. The energy source is geological, dependent on magma movement, on tectonic activity, on cracks in the crust remaining open. All of these are temporary. The crust shifts, cracks seal, magma moves elsewhere. The vent dies. Inevitably. After the vent stops, the site remains altered. The chimneys stand. The shells accumulate. The empty tubes create structure. This structure attracts other animals. Not vent specialists, but generalists. Fish, octopuses, sea stars. They use the structure as habitat, as shelter, as hunting ground. The site transitions from a chemosynthetic ecosystem to a hard-bottom community. Different animals, different energy source. They feed on marine snow, on each other, not on bacterial production. The vent is gone, but the habitat remains. Changed, repurposed, Over time, sediment covers the site. Slowly, millimeters per year, centimeters per decade. The shells are buried. The tubes are buried. The chimneys erode, collapse, fragment, get covered. Eventually, the site is indistinguishable from the surrounding plain. Flat, featureless, sediment covered. But beneath the surface, the remnants persist. Shells, mineral deposits, tubes, fossils waiting to be found. If the seafloor is ever uplifted, exposed, eroded, the fossils will reveal what was. A vent, a community, life that existed in darkness, sustained by chemistry, independent, thriving, then gone, but recorded, preserved, waiting. The mid-ocean ridge continues. Always. Plates pull apart. Magma rises. Cracks open. Water circulates. Vents form. This has been happening for billions of years. It will continue for billions more. As long as the Earth's interior remains hot. As long as tectonics operate. The system persists. Individual vents come and go. Communities form and dissolve. But the phenomenon continues. Somewhere along the ridge, a vent is always opening. Somewhere else, one is always closing. The distribution shifts. The locations change. But the total remains roughly constant. Vents exist. Life exists at them. Chemosynthesis continues. Uninterrupted. The bacteria are old. Their lineages trace back to the earliest life. Before oxygen. Before photosynthesis. Before complex cells. They have been performing chemosynthesis for over 3 billion years. The mechanism has not changed. The chemistry has not changed. Hydrogen sulfide is oxidized. Carbon dioxide is reduced. Glucose is produced. The same reaction. The same result. Repeated endlessly. Across billions of years. Across millions of vents. Unchanging. Reliable. Fundamental. Animals arrived later. Much later. But they too have been at vents for hundreds of millions of years. Their fossil record is clear. Tube worms in ancient rocks. Clams. Mussels. The same forms. The same adaptations. Time has passed. Species have changed. But the pattern remains. Animals host bacteria. Bacteria perform chemosynthesis. The partnership continues. Stable. Successful. Proven. There is no progression here. No advancement. No improvement over time. The system functions. It has always functioned. It will continue functioning. Evolution does not drive toward complexity at vents. It drives toward stability, toward persistence, toward efficiency. The simplest solution that works is maintained. Nothing more is needed. Nothing less would suffice. The system is balanced, has been balanced, remains balanced. The surface ocean covers 70% of the Earth, approximately 360 million square kilometers. The average depth is 3,700 meters. The volume is enormous, 1.3 billion cubic kilometers. Most of this volume is deep ocean. Dark, cold, distant from sunlight. Photosynthesis occurs only in the top layer, perhaps the top 200 meters. Below that, only consumption. Recycling. Descent. The deep ocean is the largest habitat on Earth. By volume, by area, by mass of water, and most of it remains unexplored. Hydrothermal vents have been studied for less than 50 years. A few hundred vent fields have been visited, documented, mapped, but the mid-ocean ridge is 60,000 kilometers long. Only a tiny fraction has been surveyed. Most of it has never been seen, never been sampled. The vents that are known represent a small sample, perhaps 1% of the total, perhaps less. Thousands of vents likely exist undiscovered. Tens of thousands possibly, each one hosting a community, each one unique in some way. Different chemistry, different species, different history, all of them operating in darkness, unseen, unknown, continuing regardless of observation. Cold seeps are even less explored. They exist on every continental margin, scattered across the seafloor, difficult to locate, no visual signature, no plume, no heat, just slow methane release, chemical traces in the water. Most seeps are found by accident, during other research, during surveys for oil and gas, during submersible dives for unrelated purposes. How many exist is unknown. Estimates vary by orders of magnitude. Thousands certainly, possibly hundreds of thousands. Each one small, each one isolated. Together they represent a significant habitat, a significant contribution to deep-sea productivity. But quantifying that contribution requires knowledge that does not yet exist. The abyssal plain covers more area than all continents combined. Flat, sediment-covered, seemingly empty, but inhabited, sparsely, quietly. The biomass per square meter is low, but the area is vast. The total biomass is substantial, and beneath the sediment, bacteria dominate. Chemosynthetic bacteria among them. Widespread, diffuse, unnoticed. Their contribution is difficult to measure, small in any given location, but cumulative across the entire abyssal plain. Possibly significant, possibly not. The data does not exist to say. The questions remain open. Studying the deep ocean is difficult, expensive, time-consuming. Ships must travel days to reach remote sites. Submersibles dive for hours to reach depth. Bottom time is limited. A few hours per dive. Then ascent. Recovery. Preparation for the next dive. Each dive costs tens of thousands of dollars. Each dive samples a tiny area. A few square meters. A few hundred meters of transect. From this, conclusions are drawn. Generalizations made. But the sample size is minuscule. The variability is high. The unknowns are vast. Remote vehicles help. They can stay down longer. Days, weeks. They can cover more area. Map larger regions. But they cannot collect samples as effectively. Cannot manipulate objects. Cannot perform experiments. Trade-offs exist. Every method has limitations. Every dataset has gaps. Slowly over decades, knowledge accumulates. Patterns emerge. But the deep ocean remains largely unknown. Most of what exists there has never been seen. Most species have never been collected, never been named, never been studied. They exist. They function. They reproduce. They die, all without observation, without record, independent of science, independent of knowledge. Some organisms are known only from fragments. A single specimen, damaged during collection, poorly preserved, enough to describe, to name, to catalog, but not enough to understand. Their biology remains mysterious, their ecology unknown, their distribution uncertain. They exist in collections, in jars, in museums, waiting for more data, more specimens, more context. That data may never come. The organisms may be rare, or the collection site may be unreachable. Too deep, too remote, too expensive to revisit. The fragments remain, hints of something larger, something unexplored. The surface world changes rapidly. Season cycle, weather shifts, temperatures vary, day and night alternate. Life responds, adapts, migrates, hibernates, blooms. Activity is tied to these cycles, driven by them, shaped by them. In the deep ocean, these cycles do not exist. No seasons, no day, no night, no weather. Temperature is constant. Light is absent. The environment is stable, has been stable for millions of years, will remain stable for millions more. Life here is detached from surface rhythms, from solar cycles, from atmospheric conditions. It operates on its own time, geological time, chemical time, time measured in the opening and closing of vents, in the accumulation of sediment, in the drift of tectonic plates. An animal born at a vent knows nothing of sunlight, has no concept of day or night, no experience of seasons. Its entire existence is darkness, pressure, cold or heat, depending on proximity to vents, chemical gradients. This is not deprivation. This is normalcy. The animal is not missing anything. It has everything it needs. Sulfide, oxygen, substrate, mates. Its needs are met. Its adaptations are sufficient. It thrives in conditions that would kill surface organisms. But those conditions are not extreme to the animal. They are home, expected, required. The idea of sunlight is meaningless here. Not absent, not blocked, meaningless, irrelevant to function, irrelevant to survival. The animal's biology does not include any mechanism to detect light, any response to light, any use for light. Light is outside its reality, outside its evolutionary history, outside its environment. The animal exists in a separate system, connected to the rest of the biosphere through shared ancestry, through shared evolutionary origins, but disconnected in function, in energy source, in environmental requirements. If you could ask the animal what it needs to survive, it would not mention sunlight. would not mention the surface, would not mention anything outside its immediate environment. Sulfide is essential. The vent is essential. Bacteria are essential. Everything else is irrelevant. The animal's world is small, meters across, centered on the vent. Beyond that, nothing matters. The abyssal plane is a barrier, not a habitat. Other vents exist but are unreachable. Might as well not exist. The animal lives in isolation, complete isolation, and functions perfectly within that isolation. Needs nothing more. Knows nothing more. Somewhere along the mid-ocean ridge, a vent is opening now. Water is heating, rising, beginning to flow. Bacteria will arrive. Larvae will arrive. A community will form. This has happened countless times. Will happen countless times more. The process is routine, predictable, ongoing. Somewhere else, a vent is closing. Flow is weakening, stopping. Animals are dying. Shells are settling into sediment, becoming fossils. This has happened countless times. Will happen countless times more. The process is routine, inevitable, ongoing. The cycle continues. Opening, closure, formation, collapse, life, death, preservation, erosion. The rhythm is slow, measured in decades and centuries, but consistent, unbroken, driven by forces that operate on geological timescales, indifferent to individual lives, indifferent to communities, indifferent to observation. The system persists, self-sustaining, self-renewing, independent. This is only one of many hidden environments shaped by isolation. Beneath Antarctica's ice, others exist that remain sealed away. In the next film, the journey continues toward the lakes hidden beneath Antarctica's ice. A world separated from the surface for far longer than the ocean below the shelves. The deepest trench is 11,000 meters. The Mariana Trench. At its bottom, pressure exceeds 1,000 bars. 1,000 times surface pressure. Animals live there. Fish, amphipods, sea cucumbers. They are not abundant. Food is scarce at such depth. But they exist. Functioning. Adapted. Surviving. No light has ever reached the bottom of the trench. No photosynthesis has ever occurred there. The food comes from above. Marine snow. Falling. Slowly. Most of it is consumed before reaching such depth. But some arrives. Enough. And in the sediment, bacteria perform chemosynthesis, breaking down what organic matter exists, oxidizing what chemicals are available, producing small amounts of glucose, supplementing the meager input from above. The ecosystem functions, barely, at the edge of viability, but functioning. At such depth, water behaves differently. Its density increases under pressure. Its viscosity changes. Sounds travel faster. Chemical reactions proceed at different rates. The physics are altered, not dramatically, but measurably. Life adapts to this too. Proteins fold to account for the pressure. Membranes adjust their composition. Enzymes maintain function despite the altered kinetics. Metabolism continues. Slowed, modified, but continuous. There is no bottom to adaptation. No depth where life cannot exist. Only depths where life becomes increasingly sparse, increasingly specialized, increasingly detached from surface systems, but never absent, never impossible. As long as water exists, as long as organic matter exists, as long as energy is available in any form, life finds a way, not through intelligence, not through intention, but through variation, through selection, through time, billions of years of time, enough time to explore every possibility, to fill every niche, to adapt to every condition, however extreme, however isolated, however dark. The ocean is deep, deeper than light can travel, deeper than warmth can reach, deeper than most life can tolerate, but not deeper than all life, not deeper than chemistry, not deeper than possibility. At the bottom, in complete darkness, in crushing pressure, in near-freezing cold, life exists. Simple, sparse, slow, but present, functioning, continuing, and it will continue long after the surface changes, long after species rise and fall, long after continents drift and oceans shift. The deep will remain, dark, cold, stable, and life will remain in it, adapted, persistent, quiet, operating on principles that predate sunlight, on chemistry that predates life itself, unchanged, unchanging, separate. The vent flows, the bacteria synthesize, the tube worm extends its plume, absorbs sulfide, feeds, grows, slowly, patiently, in darkness that is absolute, in darkness that is normal, in darkness that is all there is, and all there needs to be.